All right, we're going to finish up this metabolism video by looking at the process of fermentation and then the generation of ATP through aerobic respiration using a non-carbohydrate source. Let's go back to our slides. Okay, so fermentation is an incomplete oxidation of those carbohydrate molecules in the absence of oxygen. So when an organism requires a rapid production of ATP, they don't have the time essentially, or when a cell needs to produce ATP and needs to do it very rapidly, and there's very low oxygen availability or none, and instead of aerobic respiration, they will switch over to a fermentation process. Okay. This concept here is the organic compounds, either lactic acid or an alcohol, are used as terminal electron acceptors. So for all of these things, just think in biology and biochemistry specifically, there's a balance that must be attained. We've already talked about the ATP to ADP cycle, where ATP is used essentially to power reactions, but that ADP needs to be phosphorylated to make ATP back and forth and back and forth. We saw that also with these electron carriers, that NAD can be reduced to NADH plus H plus, but that also needs to be oxidized back again. So those molecules must be continuously recycled in the cell. So fermentation, because this is just a super rapid partial oxidation of glucose through glycolysis to generate a massive amount of ATP, there's a problem that there are these reduced electron carriers generated, and there's no way for them to go downstream and donate them to the electron transport chain. So fermentation is essentially just taking that pyruvic acid end product and then converting it into an alcohol or an acid and allowing those organic compounds to accept those electrons from the reduced electron carriers. This is an anaerobic process, right? So this can happen anaerobically. And there are two major categories here. It's either alcohol or acid fermentation. But again, just put conceptually in your head, the whole point here is to allow those reduced electron carriers to be regenerated, to be recycled back into their oxidized form by the donation of those electrons and protons, either directly to pyruvic acid to form a lactic acid here. That would be acid fermentation or homolactic acid fermentation. This is what our human muscle cells do, when we overpower them and we don't give them enough, starting ATP, and they need to switch over. They begin to ferment and produce lactic acid by taking in glucose and just very, very rapidly converting it to pyruvic acid and then to lactic acid, which is a waste product that's released outside of the cell. And tons of bacteria do this too. All the lactobacillus, for example, that are in your yogurts, your dairy foods, those are all undergoing this lactic acid fermentation process. Alternately, a cell like many yeast organisms will take glucose, convert it to pyruvic acid through partial oxidation and glycolysis, generating those reduced electron carriers, and then that pyruvic acid can be partially oxidized to acetaldehyde, which then can receive those electrons from that reduced electron carrier, NADH plus H plus, to produce an oxidized NAD and a reduced alcohol. In this case, it's ethanol, right? So this is the generation of an alcohol. And then for both acid fermentation or alcohol fermentation, both of these organic end products can be further modified. We've already seen an example of this in the laboratory with mixed acid fermentation, where lactic acid is essentially continuously modified to produce these stable mixed acids, generating a whole bunch of, lowering the pH quite substantially. Alternately, there are other alcohols that can be made. Really, any alcohol you can think of that you know of as an alcohol solvent, they are generated through fermentation and a continuous conversion of that final fermentation product through alternate pathways. We looked at this one in VP test, so that 2,3-butane-diol fermentation, producing acetone, which is a neutral end product of the alcohol fermentation. Right, so we've seen examples of this. There are many, 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 many other examples of what organisms can do during their fermentation pathway. But these are just some. We know that these products of fermentation are captured by humans and used, and it's really cool actually if you are a historian or history buff, then to look at essentially how humankind figured this out. Almost everything is by accident, but many of these fermentation products are accidental fermentation of grains that were left in a silo and it got wet, and then organisms fermented the stuff and then it produced these amazing compounds that got people drunk. So you can go look at that if you want. But everything you see here are all products of fermentation, including vitamins, antibiotics, and hormones that are used by biotherapeutic industries often. What's the energy yield? Well, it's just what's produced in glycolysis. Therefore, you know that there are 4 molecules of ATP generated altogether, 2 used, so a net total of 2 ATP generated through substrate level phosphorylation for fermentation, but aerobic respiration is 38 molecules of ATP. So if an organism has the ability to, or if a cell can, it will undergo aerobic respiration preferentially. It's a much higher energy output for every input of glucose, but in the absence of oxygen, or when ATP is needed very, very rapidly, then that cell can switch over to a fermentation pathway to just essentially kick out a ton of ATP through glycolysis. And there you go, just answered that second question. How do they make up for this low energy production? They do it very, very rapidly. This question, fermentation is often used as a food preservative. I had you guys work through this problem in class. Explain why it would slow down food spoilage. One of the most basic things to think about is, what is the product of fermentation? Well, it's either an alcohol or an acid. So very low pH, or even potentially, yeah. In both cases, we are dropping the pH with the production of CO2 for alcohol fermentation, or production of acid, lactic acid, or other acids for acid fermentation. And so we know that organisms are super sensitive to alterations in pH in their environment. We saw that already in our environmental lab. And because that change in acidity can alter their enzyme function. So that alone would definitely help reduce food spoilage, the drop in pH. But also think that this is a super rapid process, and that initial glucose or carbohydrate that's in the food will be used very, very rapidly by organisms that are fermenting, which is why cucumbers taste very sour when you pickle them or when you allow them to ferment, because all of that sugar that's in that starting cucumber, that whatever that thing is that you're putting in there, or any of the kimchi products that are on the market, they all have no sweetness to them. There's none, because all of the sugar is gone and used by those fermenters, which makes it actually quite difficult for other organisms to live there, because now there's no carbohydrates available, and the environment is quite toxic to the organisms. Pretty cool concept. This is an example of a discussion question that you definitely will see on the exam. So I'll just read through it first, and then we'll go over it all together. Explain the following observation. Cells of E. coli fermenting glucose grow faster when NO3, which you guys know is nitrate, is supplied to the culture, and nitrite is produced, and then even faster and stops producing nitrite when the culture is highly aerated, which means oxygen is provided to the culture. So note that anytime you see a couple of key things here, I wonder what color this is. This is red. Hold on just a second. Okay, a couple of key things here. Anytime you see this concept, grow faster, here, changes even faster. This always means change in metabolic pathway, okay? Anytime you see a change in growth rate, then we know that we are looking at, let me change this to white, I think that'll be easier. Just so you know, change in growth rate is usually, whoops, sorry about that. Usually, not always, but usually indicative. It indicates indicative of change in metabolic pathway. Okay, so if you see a change in growth rate, like grow faster, grow slower, stop growing, whatever it might be, that usually means something's happening metabolically, and you should pay attention to whatever else is discussed to try to figure out what actually is occurring at the biochemical level, okay? Secondarily, as you're looking through something like this, you really want to identify some of the key things that you're noting, right? So when you look at this, NO3 is supplied and NO2 is produced, you should really be thinking about what that means to you. When NO3 is available, which is nitrate, and NO2 is produced, we already know this is through the enzyme nitrate reductase, but I just told you that this is also an end. Whoa, didn't mean to do that, sorry. This also can be a terminal. Why is it doing that? I think I lost my pen. This also can be an end. Ah, oh no. An end, no. Sorry guys. I definitely lost my pen. This can also be an end, there we go, electron acceptor in the process of anaerobic respiration. Okay, and then I also, the last thing I want to note before we go through this step by step, one more thing to note is that organisms preferentially, if they're given the opportunity, will increase or change their metabolic process to the most energy favorable process, right? Orgs will use the most energy efficient process. Just think about that. They'll use the most energy efficient process at any time possible. Okay, with this information, we should be able to answer these questions. What metabolic pathways can this organism undergo? Or you can consider this metabolic processes like aerobic respiration, anaerobic respiration, fermentation are the three processes we're asking about. And then what oxygen classification? This comes back from chapter 6 on our chapter on nutrition. We're looking at all of those specific oxygen classification categories. Okay, so let's answer this one first. We got to figure out, let's answer the first one first. What metabolic pathways or processes can they undergo, considering all the stuff we looked at? So let's start at the top. Explain the following observation. Cells of E. coli fermenting glucose stop here. Let me change my color so we can highlight a couple of things. Okay, fermenting glucose. So we already know that these organisms can ferment, right? So we know first, we can say fermentation. And how do we know? We know this because I told you. Because it's stated. Now, I could alternately tell you, you had the following observation in the laboratory. You inoculated a glucose deep and you saw it turn yellow. That means you would have to understand or remember from your biochemical testing that glucose deeps, right? These are agars that have, red pH indicator, and yellow means that it's acidic, which means it's indicative of glucose fermentation. So think along those lines. I'm making it easy for you right now, but really be prepared to take your knowledge from laboratory of different biochemical tests and use the results of those biochemical tests to answer this question too. But in this case, I just told you about fermentation. Let's keep going then. So cells of E. coli fermenting glucose grow faster, and I just told you this means something different is happening to their metabolic process. So let's see what they're switching to. When NO3 is supplied to the culture and NO2 is produced. Now again, I go back up to the top note, and I just told you that this process that we have already looked at in the classroom is showing you that anaerobic respiration is occurring as nitrate is the end electron acceptor in organisms, in some organisms with this capacity, with that nitrate reductase enzyme. So the second part here that we just saw, right here, where NO3 is supplied and NO2 is produced, means that it can undergo anaerobic respiration. And we know this because we see the nitrate is reduced to nitrite, and this process this process is indicative of anaerobic respiration. And then let's keep reading, and then even faster. Again, this means some change to the metabolic process and stops producing NO2. This is also keenly indicative that there is no more anaerobic respiration occurring when the culture is highly aerated. Aerated means the presence of oxygen. So if they're no longer undergoing anaerobic respiration and they're growing faster, which means they're changing to a more energy efficient process, then we know that this means that they can also undergo aerobic respiration. And we know this because they grow faster. Sorry. We know this because they are growing faster. Faster. Sorry guys. Growing faster. Nope, that's not what I want. Growing faster with oxygen. Oh my gosh, I'm getting challenged here. Growing faster with oxygen. Cow. Growing faster with oxygen. So if they can undergo fermentation, anaerobic respiration, and aerobic respiration, if you go back to that table that we made from chapter 6 on oxygen classification, you should have figured out this is a facultative guys, facultative aerobe or anaerobe. Okay. A facultative anaerobe. There it is. Okay, done with that question. Okay, there are two more things that we need to just discuss. And sorry for all these kids coming in and out. It just got sunny out here, so they're coming out a lot. If an organism does not have carbohydrate like glucose or sucrose or lactose, they can still live by breaking down their own lipids from their cell membrane or from endosomes that are on the inside of their cell, or from lipids that they can make themselves. So this is through either lipase activity, where enzymes are key enzymes that actually break apart fatty acids to produce metabolic intermediates like DHAP that can step into the glycolytic pathway and continue that oxidation reactions that we saw before, or beta oxidation, which is a process that our cells undergo when we are in ketosis or using ketones or using lipids to generate ketone bodies and also generate energy. So beta oxidation is a process I want to review with you guys because it's a really great overview of everything that we just did. So in beta oxidation, a fatty acid compound, which is a hydrocarbon or carbons and hydrogens that are all bound together with either single or double bonds, can be broken down through very key enzymes to generate acetyl groups, 2 carbon acetyl groups that are added to a coenzyme A. And then this fate of the molecule, acetyl coenzyme A, is exactly the same as we've seen before, where that acetyl group is donated to oxaloacetate and then can shuttle the turn of the Krebs cycle one time. So let's do a quick rundown of how this process can occur with a 6-carbon fatty acid. Those of you who are biochemistry people, you'll tell me that what I'm telling you is wrong, but that's okay because I'm just doing a very brief rundown of this process here. So let's see how this works. This is from my last video where we saw the whole process of aerobic respiration. I'm going to clear a part of this thing, so bear with me. I'm going to erase all of this stuff over here, okay? So that I can write this whole thing out using pathway that we wrote out on the longhand version. Now I do have a brief video of this one already because I wanted to explain the process of beta oxidation for an in-class discussion question I have for you guys, but let me just review it again one more time here. Give me just a second. This is impossible. Okay, beta oxidation. Here we go. Okay, so beta oxidation is what we're drawing out here. Let me sort of give us a little space. Beta oxidation is the process where a fatty acid compound, and I'm going to draw a 6-carbon one like we have in our example, 1, 2, 3, 4, 5, 6, where 2 of the 6 carbons in our example, or 2 carbons of a fatty acid molecule, are donated to a coenzyme to form acetyl coenzyme A. Now there is a bunch of stuff happening here. So we have a coenzyme A, of course, needs to come and pick up those 2 carbon compounds to form that acetyl coenzyme A. There is a reduction of 1 molecule of NAD. And there's a reduction of a molecule of FAD. Okay, now we have this acetyl coenzyme A generated, and now that you can see the stuff up here, note that this acetyl coenzyme A is going to have the same, right? I'm going to kind of circle this molecule. Acetyl coenzyme A is essentially going to go right there and have the same fate as this acetyl molecule. Right, so I'll just rewrite it for you, but note that it's going to be exactly the same as we see here. So this acetyl group from the acetylated coenzyme A will be donated to oxaloacetate to power the Krebs cycle, a single turn of the Krebs cycle, because we only have 1 acetyl group, right? That acetyl group will be donated to that 4-carbon oxaloacetate to make that 6-carbon citrate. So that happens only 1 time for every acetyl-CoA that's generated here. So we have 1 turn of the Krebs cycle. Let's draw very quickly our energy yields that we know come off of the Krebs cycle. First, we know this is a complete oxidation of this acetyl group, so there are 2 carbons in that. Therefore, there's a loss of those 2 carbons and the process. There is ATP generated here, so we have 1 ATP for each turn of the Krebs cycle. There are reduced electron carriers, so we have 3 for each turn of NADH. And we have 1 FADH2. All right, so now let's count up all of our energy yield and potential and actual energy yield. So here, let's start with our how many NADH plus H plus molecules are generated. We have 1 in an actual beta oxidation event, and then 3 in the single turn of the Krebs cycle for each beta oxidation event. So altogether, 4 NADH plus H pluses. For FADH2, there's 1 generated in this beta oxidation event and 1 generated in 1 turn of the Krebs cycle from that acetyl group. So we have 2 FADH2s generated here for each single beta oxidation event. Let's go and remember our conversions here. So for every NADH plus H plus, we have the potential to make 3 ATP molecules through oxidative phosphorylation. 4 times 3 is 12 ATP through oxidative phosphorylation. For every FADH2 entering into the electron transport chain, this has the potential to make 2 ATPs, which generates, right, for each beta oxidation event, coupled with the turn of the Krebs cycle, generates upwards of 4 molecules of ATP, again through oxidative phosphorylation. Now don't forget we have 1 ATP that's generated through substrate level phosphorylation in the Krebs cycle. So let's add that here. So for each beta oxidation event, we have 12 plus 4, which is 16, plus 1 is 17 molecules of ATP for each per beta oxidation. Right, so for each of these 2 carbon compounds, that generates an acetyl group that is coupled with coenzyme A, turns to Krebs cycle, and all of this reduced electron these reduced electron carriers coupled with that substrate level phosphorylation can generate 12 molecules of ATP. The last thing we need to figure out is how many beta oxidation events can occur here. We're going to keep it simple and just say, well, let's just figure out how many sets of 2 we have. There was 1, there's 2, and there's 3. Or in other words, 6 carbons divided by 2 carbons for each beta oxidation event makes 3 events, or 3 reactions that can occur. So we'll take this final number, multiply it by 3 to give you 51 molecules of ATP for that 6 carbon. Okay. So I just walked you through how it's possible to generate about 50 molecules of ATP through the 6 carbon fatty acid compounds. So this is an energy-rich process, and this was an in-class discussion question I have for you guys. If you haven't done it yet, you still have a little bit of time to get this up on Canvas. So please make sure you complete this exercise. This is just talking through the whole process of beta oxidation again. You don't have to memorize this process, but I do want you guys to be able to use your understanding of metabolism, understand that a cell will oftentimes use non-carbohydrate sources to generate ATP, and this is just one example of how that can occur. And it really just takes advantage of your knowledge of that basic metabolic process and the potential energy that each of those steps can generate. As mentioned, proteins can be an energy-rich source also for a cell, and sometimes not just proteins, but just peptides or peptones for partial digestion of proteins. So there are proteases. We've already looked at a few examples of that in lab. We looked at gelatinase and urease as examples of proteases that can break down these larger proteins, producing amino acids, some deamination reactions that can actually remove that amine group and make a hydrocarbon that is available for energy use. So that this picture here is showing you a deamination reaction where glutamic acid is deaminated to make alpha ketogluteric acid and an amine group. That alpha ketoglutarate can enter into the Krebs cycle. I've already talked to you guys about that, and it can essentially be partially oxidized to generate more reducing power that can then go and power ATP in aerobic or anaerobic respiration. So there's definitely energy production that can occur through the use of proteins here. Our cells do also undergo this process in the absence of carbohydrates or sometimes in other processes in the cell. So there's lots of different examples of this, but a really common example, there's a few different steps in this reaction, but I've summarized it in this little reaction here. So alanine is a very simple amino acid. It can be deaminated essentially through a couple of steps in this process and generate pyruvic acid. This is coupled with a reduction event where an NAD is reduced to NADH and the pyruvic acid is generated. So the question here is how much ATP can be generated from one alanine molecule through aerobic respiration. Let's see if I have any more room on that whiteboard. So let's try to draw a little line here and do this last example down at the bottom. Where did my pen go? Here we are. Okay, so let's do this last example here where we have an example using alanine. So in this case, alanine is converted into pyruvic acid or pyruvate, only one of them. And this process generates a reduced electron carrier. And the question is asking how much ATP can be generated through aerobic respiration from one alanine molecule. Well, let's see, pyruvic acid, and just as I drew before, let me just show you where this would fit in. Pyruvate, if we're talking about aerobic respiration, that means we're gonna talk through from here down, right? So you're gonna avoid glycolysis, because that's not occurring. Whoops. But let's see, from pyruvate or from pyruvic acid, we know we have the transition step to generate acetyl coenzyme A. This just runs through the Krebs cycle. How many times? Only one, because we only have one pyruvic acid. One turn of the Krebs cycle. We have a bunch of stuff happening here. So there is a reduced NAD, just one. Here, there are reduced NADs here, 3 of them for every turn of the Krebs cycle. There is reduced FADH2, also generated in one turn of the Krebs cycle. There is ATP generated from one turn of the Krebs cycle. And we can draw our other little things that we know are happening here. We have our addition of our coenzyme A, of course, coming in. And we have our loss of CO2s. One CO2 here, and 2 CO2s here. All right, now let's count everything up. So in calculating energy yield, let's count our NADH plus H plus. We have 1, 2, 3, 4, 5. So we have 5 of these guys here. Let's count up our FADH2s. There's only one FADH2. And let's count up our ATPs. Again, we have 1 ATP here. Sublevel phosphorylation, and let's add our conversions here. So for NAD, we're multiplying this by 3 to make 15. For FAD, we're multiplying this by 2 to make 2. We'll carry over our ATP through substrate level phosphorylation, and now we have 15, 16, 17, 18. So for every alanine molecule that is deaminated to make pyruvate, with the potential to generate 18 molecules of ATP through aerobic respiration. Hopefully this makes sense to you. Okay. And that's it for catabolism. For anabolism, I just wanted to review a couple of concepts here. Anabolic reactions, remember, are biosynthesis. It's always building up macromolecules from simpler compounds. In particular, we just looked at a bunch of ways that precursor metabolites can be generated through catabolism, and they can themselves be used for biosynthesis reactions. These are the 4 basic building blocks we remember: carbohydrates, amino acids, fatty acids, and nucleotides. For cell, for biosynthesis, these basic macromolecules need to either be acquired from the environment or synthesized inside that cell. Synthesis doesn't have to start from the DNA level. We just saw examples of how precursor metabolites can be synthesized through carbohydrate catabolism, for example, right? Or deamination reactions from amino acids to generate other intermediates there. So there are different ways to generate these intermediates needed for biosynthesis reactions, and these are a couple of them. All pathways are varied. There are specific enzymes that catalyze each step in each of these biosynthesis pathways, and all of them require the input of ATP because we are forming bonds. And to sort of close this up, so we've just looked at how catabolism is breaking down compounds through oxidation reduction reactions, generating ATP and reduced electron carriers. But then all the way through, we talked about these precursor metabolites or metabolic intermediates that can be pulled out at any time. So if the cell will pulls these compounds out and uses them for biosynthesis, for example, the production of amino acids, or the synthesis of amino acids for the generation of proteins, synthesis of different carbohydrates for these larger components, even the synthesis of fatty acids for the basic ingredients of lipids or fats for membrane. All of these things can be generated from these catabolic metabolic intermediates that are generated here. And then alternately, we just saw examples of beta oxidation where a fatty acid can be used to produce energy, so a catabolic reaction from fatty acid, or we also saw how alanine or other amino acids can be deaminated and then be used to power the Krebs cycle or to be put back into these respiration pathways. So this kind of back and forth between these central catabolic pathways or metabolic pathways and these different biosynthesis reactions is called amphibolism, where there's a complete integration of catabolic and anabolic reactions. So it's not as simple as just looking at ATP being produced from catabolism that can be used to generate the power for anabolic reactions. Now I really want you guys to just keep in mind that it's all these metabolic intermediates too. And then just to give you a little bit of relief, understand that you don't need to memorize this. This is a better picture of the immense complexity that's happening inside the cell at any given moment. This is nice to know that you actually can recognize what's happening here. This whole central part is glycolysis. Here's acetyl coenzyme A, so pyruvate to acetyl coenzyme A, and then the turning of the Krebs cycle is here. So just so you're aware, like these are the processes we just looked at, but this is the reality of the cell, that there are so many steps along the way that the cell can either shuttle things into or pull things out of these catabolic reactions for biosynthesis or vice versa, right? And that is the end of our metabolism stuff. Thanks for listening.